No, thank you. So this was the title I was given, and I've taken the liberty of changing it a little bit to this, partly because I didn't want to just cover what had been covered before, but equally I do want to say a few things that relate to other talks. So I thought I'd choose this more generic title because it will allow me to speak about three things that I think are interesting and we haven't explored fully. So one of those is I want to talk about the genetic influences on pain. So how your particular sequence of DNA that you have, and that's obviously different from my DNA sequence, one base in every couple of hundred will be different, how your genetic variants and mine may affect our pain sensibility. Secondly, I'd like to address, really it's a follow-on from Per's talk, and that is how in chronic pain states, alterations in gene expression, so not your gene, not your sequence, not the genes you have, but how they're expressed, how they may play into the pain state, and that will be unashamedly a study of what we can learn from animals, and I'd hope to rebut or at least qualify some of the discussion that we had before, and again I'd welcome any interruptions. And then thirdly, I thought I'd tell you about something we've been doing with genes recently in the context of sex differences. I anticipate that I'm going to run out of time in this lecture, so if I don't get to number 3 tonight, then maybe I'll just push that into tomorrow's lecture. Okay, so I want to start by saying something about how genes in humans may affect the pain that they feel. And the first point to make is with this this figure, which I don't know if you can see. It's a Mauritian fakir. This was a slide given to me by an old-time neurologist, Peter Nathan, who's long dead, but he studied this fakir in London. And if you can see, this fakir is doing, he's doing lots of odd things, including the way he's dressed, but he has a 4-foot épée through his tongue, another one through his abdomen, a couple of daggers casually shoved through. This one must be quite close to his carotid, actually. So this is what he did for a living. And the point was that he claimed that this was completely painless, this fakir. And so I only show it to illustrate the point that we're actually quite different in how we feel pain. And you can study that more systematically in a number of ways. So here's a little bit of data, and there's a lot out there emphasizing this diversity in pain sensitivity. So this experiment on the left is a very simple one. You take a bunch of people, it's a small group of people here, although we've done this experiment with 2,000 people now, and you see exactly this pattern. You take a bunch of people and you apply a hot probe, a 49-degree probe to their skin, and ask them to rate that stimulus. And they're rating it on a VAS scale from 0 to 10. And you can see that these 16 subjects, some of them report very low levels of pain and some very high levels. And I picked this data set to illustrate the diversity in pain sensitivity because this experiment was done in a scanner, so you could ask at least whether or not the physiological activation of brain circuits was linked to their reporting. I mean, one problem, to throw it back to Per a bit, one of the problems with studying pain in humans is that sure, we have no measure, we have no objective measure of the pain they feel, so we have to ask them. So whilst you might complain about the outcome measures we use in animals, don't forget in humans you're depending on their subjective report, which again has never been validated with anything because there's nothing to validate it against. Anyway, this says that whatever they're reporting was going along with, at least in some brain areas, brain activation patterns. And here's a second experiment from the animal world, actually, coming from the early work of Jeff Mogul. And Jeff had the clever idea of studying inbred mouse strains. So mouse, I don't know if this is true, but the story is that mice were kept as pets in the Victorian era in several countries, and so they were all inbred, the way that some dogs are. And so we have mouse strains that have quite distinct genetics. And you can ask whether all of those different inbred strains are equally sensitive to pain. So this is a test of how much you have to prod their hindpaw before they decide they don't like it enough to withdraw. And you can see that some animals are very sensitive down here, and some are much less sensitive. And I would say, as an aside, that you ought to be careful about all of the pain studies that use transgenic animals in which genes have been manipulated, because the traditional way of making those animals is to cross this strain with this strain. And you'll notice you're crossing a very insensitive with a very sensitive strain, and that can lead to lots of uncertainty about what the hybrid strain should look like. Anyway, the key point is that the mouse strains are different, humans are different, and that may at least, one of the factors that's likely to contribute to that difference is genes. So another way of saying what I've just said is that if you took a large population of people, and that this has been done, you find that pain sensitivity is sort of normally distributed with quite a broad bell-shaped curve. But actually this is not quite the full story. And not to scowl, it turns out that there are very small numbers of people at this end and at this end of the spectrum. And I think this end of the spectrum has attracted most of the interest of pain scientists in the last decade or so, because it represents people who essentially are totally insensitive to all forms of pain. And they're an interesting group, because at this end and this end, we have identified a couple of single genes that are mutated in individuals that give rise to this loss or gain, almost total loss of function, or a pathological gain of function in pain sensitivity. And there was a question asked earlier about whether single genes mean anything. Well, here there are good examples that manipulations of one gene so that it changes its function is enough to completely change an individual's pain sensitivity. And obviously, from a therapeutic perspective, if you could reduplicate this phenomena with a tablet, you might have a very good analgesic drug, because for the time that the drug was on board, you'd make people insensitive to whatever it was that was causing their pain. This bit in the middle is a bit more tricky to explain. It's certainly not all genetic, but I'll talk about these bits for a few minutes, and then I'll talk about what we know about the genetics contributing to this sort of normal pain sensitivity. So first of all, just to reiterate the slightly amazing feat, given the evolutionary conservation of pain across a large spectrum of animals, as you heard yesterday from Tony, from Aplysia to us, you'd think that pain has a fantastically important survival value, and it does. So people who have this condition have a shortened life expectancy. But it is sort of amazing is that these individuals that can be classified in a couple of slightly confusing ways, the key points are that they don't respond to acute tissue-damaging stimuli. There's not many of them, perhaps 1 in a million, so they haven't had the full gamut of slings and arrows that the real world throws at the rest of us. So we're not quite sure about whether there are exceptions, but for the most part, they don't have any pain with chronic injuries. So we've studied one woman in South London, had a completely painless childbirth. They don't have pain with broken legs, they don't have pain. They don't have migraine. There's a bit of an argument about whether they feel itch, actually. Some of them don't sweat. Some of them seem to have some cognitive impairment. Some of them have funny immune dysfunctions. So these last two factors are not fully understood. But the important point is, it's not that they lose pain because everything is messed up. They have relatively or completely normal other sensory and even motor functions. So there's something very selective about this loss. So what causes it? Well, we have 3 or 4 genes that have now been identified that in a Mendelian way can precipitate this syndrome. So the first one identified was a failure in the signaling between this trophic factor, nerve growth factor, and its receptor. And we know from a lot of developmental biology that NGF is essential for the normal development of the small diameter peripheral nervous system and the sympathetic postganglionic system. So a long time ago, we made an NGF knockout animal, and that has the same phenotype that you see in some of these patients who don't feel pain. That is that these mice, they lose all of their small diameter sensory neurons. So if you look in the DRG, these black blobs are the sensory nerve fibers. Most of them are gone. If you look in detail, it's about 70%, and specifically, it's the small diameter, 70%. And this phenotype is seen with a loss of NGF function, if that's mutated so that it can't do what it normally does, or a loss of its primary receptor, known as TrkA. And in fact, we now have individuals with, most of the individuals have mutations in TrkA, few of them have mutations of NGF. So that's terribly interesting. It's associated, if you take a biopsy of a peripheral nerve, it's a sural nerve biopsy, even at this LM value, photograph level, you can see that the small caliber fibers are lost from their peripheral nerves. So that's terribly interesting. In fact, I was going to talk a bit more about NGF tomorrow, because the story around NGF is not exclusively a developmental one. There's an adult story which has led to quite an exciting development in novel analgesia development. But I think that this particular this role of NGF TrkA signaling in development, whilst it's biologically interesting, it in itself is not therapeutically very interesting, because of course by the time somebody gets to an adult, they don't have this dependence on NGF for survival. So we can't exploit this for treatment. On the other hand, the other mutation that's been well studied that causes congenital analgesia is a mutation in the gene that encodes this sodium channel alpha subunit. The gene is known as SCN9A. And loss of function mutations in this alpha subunit lead to, as you heard earlier from Alain, a loss of pain sensitivity, a total loss of pain sensitivity, with the only side effect being a loss of smell. And you can see that some of these mutations, the mutated gene has been cloned and inserted into some cell line and studied, and some of them at least show, as you might expect, an inability to carry inward current. That's why they don't work. The actual, at first, when people tried to model this in animals, in mouse mutants, they got a very different phenotype, and there was a lot of anxiety about whether mice and rats and humans are completely different, and da-da-da-da. All of that turns out just to be methodological. There is a very nice global knockout that's been made by Amgen and published. A couple of papers are published on this now. It was difficult. Because the animals, like the humans, can't smell, they don't feed properly, so the pups don't feed when they're first born, so you have to make you have to be careful in how you breed them. But the animals on all behavioral tests go to cutoff, pretty much like the humans do. It's interesting, when you study the animals physiologically, this is responses from a skin nerve preparation. Sure, some of their nociceptors are not working, and the ones that are working much less efficiently than normal, but nonetheless, the animals have nociceptors that peripherally can respond to increasing forces and heat. So that tells us that we don't really understand why 1.7 knockouts are painless. It's not because they've lost all peripheral input. It must be for some other funny reason. But nonetheless, this would be, if we could emulate this phenotype with a drug that blocks 1.7, you'd think we'd have a very good analgesic on board. For what it's worth, these patients, although they can't feel pain, many of them, as they grow older, they learn to kind of attune themselves to other subtle sensory signs. So here's one patient we studied that had a particular mutation. He does have he seems to have a sort of a loss of some sensory innervation, a bit of a neuropathy, but what you won't be able to see on this slide is that when we increase skin temperatures, he can guess how hot it is. He doesn't feel pain, and he's had lots and lots of injuries because of the loss of protective value, but he has some other sign, some tingling he calls it, that he can tune into, that he can use to protect himself a little bit. So that's NAV 1.7. At the other end of the spectrum, we have a few genes that lead to a gain of function. So here's a woman with erythromelalgia, this unpronounceable disease. It too arises from a mutation in the same alpha subunit, but in this case it's a gain of function in this subunit so that the channels are activated more easily than normal sodium channels. And that leads in the long axons, and why it's particularly the long axons is not understood, leads in the long axons to ongoing activity, which is perceived as spontaneous pain, a lot of the local neurogenic inflammatory signs associated with that, such as these red feet. And it's interesting that the same gene crops up as causing both loss of functions and gain of functions. So that I'm sure there'll be other people out there with interesting mutations that put them at this end of the curve or this end of the curve. We have, there was a description about a year ago in Nature Genetics of a mutation in this gene, PRDM12, leading to congenital analgesia. This is a transcription factor. I mean, they didn't show it implicitly, but I bet these people also have a failure of development of the peripheral nervous system. I bet this is going to be a transcription factor that regulates the development. And there's a claim out there that a gain of function mutation in another sodium channel, NaV1.9, can also lead to this phenotype, although I think the evidence there is that the phenotyping of the patients is much less clear-cut. And similarly, we've got quite a number of gain of function mutations in channels, including these sodium channels and this TRP channel, that can lead to this gain of function. So that raises the question, so these Mendelian genetics can have this profound effects on pain phenotypes. What about this big hump in the middle? Do we have evidence that individual genes may have a small impact on whether you're here on the curve or here? And the concept is that there are probably many genes, dozens or hundreds of genes, which subtly make you more or less sensitive to feeling pain. So how do we know that, or what do we know? Well, there have been a number of candidate genes, we've heard about one or two at this meeting, where people have looked at individuals who have different variants of a particular gene, and they've asked whether carrying that variant makes people more or less susceptible to a particular form of chronic pain. And that's not been bad. The problem is that the candidate gene studies are usually underpowered and they often have not replicated. But nonetheless, even though at the moment we don't have many good examples of the genes that contribute to this, we're pretty confident that there must be genes. And we're confident from experiments like this, experiments on twins, identical twins. So we and others now have done a number of studies. We've got a large twins unit at my institute, and so we're able to study monozygotic twins who have essentially 100% genetic identity and compare them with dizygotic twins that, like most other siblings, share about 50% genetic identity. And the idea is that if a trait like pain sensitivity is controlled significantly by a series of genes, identical twins will be more like each other in that trait. They'll be more similar to each other than dizygotic twins. And so we've chosen to study these twins with a whole number of experimental pain stimuli, and we chose experimental pain stimuli because we didn't want to study the genes that might underlie a disease state, like arthritis. We wanted to study the genes that contribute to pain. And we studied a whole series of pain traits, things that are, acute pain sensitivity, and also tests that capture some of the phenomena we've been talking about, peripheral and central sensitization. The actual data you get is not very exciting. So here it is for one trait. This is heat pain threshold. So with monozygotic twins, all you do is you plot twin 1's sensitivity against twin 2, and you get this scatter plot. And you do the same for the dizygotic twins. And what you're supposed to see here, that these are better correlated than those. And that's it. You have quite a sophisticated structured sort of equation modeling to identify how much heritability would explain this level of correlation versus this level. And so there's a whole series of pain traits here. I'm not sure if you can see them all. We've studied quite a lot, including heat pain threshold, some pain to antiphoresis of some things, secondary hyperalgesia as a measure of central sensitization. And maybe this is the crucial point, is that nearly all of these traits are significantly heritable with about 30 to 60% heritability. So that's good. That tells you that in this experimental setting, 50 or 60% of the difference in pain variability between us will be explained by genes. And again, it's a table, I'm happy to make these slides available. It's a table that just makes the point. This is not some weird thing about experimental pain. If you look at heritability across a whole number of different conditions, you end up with these levels of heritability, which are pretty concordant between 30 and 50, 60%, whether it's experimental pain, fibromyalgia, lower back pain, migraine, always end up with pretty much the same difference. And so, there's lots of evidence that there will be genes that have an important impact on individuals. Identifying those genes has actually turned out to be extremely difficult, and I'm not convinced that we're ever going to get very far in the pain field. The best data set that exists is this one that was published last year, and it relates to migraineurs. So the idea is you take a whole bunch of people, and it turns out that it has to be a very, very large bunch of people, like tens of thousands of people. You have to be able to phenotype them properly. Well, that's not so bad to phenotype a migraineur. You can probably do that on a questionnaire. But I think for most other complicated pain traits, it's difficult to phenotype people. So the idea that you're going to phenotype 20,000 people is actually it's a very improbable and difficult process. Anyway, you phenotype the people, you then sequence their entire genome, or you get an estimate of their entire genomic sequence, and then you ask whether there are bits of the genome that are associated with their particular trait. So in this case, being a migraineur or a non-migraineur. And then you do statistics to find out whether it's likely that so many people with migraine will have this genetic variant. So this kind of analysis is called GWAS, genome-wide association. You're associating their genome with a phenotype, and you get this so-called Manhattan plot, which is a flat line. So each of these vertical lines is a genomic position, typically a gene in the genome, and you're asking what's the probability of a variant in this region being associated with, in this case, migraine. And you can see that there are multiple regions where there's a whole series of dots spread up the graph representing very high levels of probability. So we have identified, and this was a kind of a meta-analysis from lots of other studies. It was 60,000 migraineurs and 300,000 non-migraineurs, and it identified 44 SNPs, single nucleotide polymorphisms, differences in genomic sequence that were associated with being a migraineur. So that sounds great. We've nailed it. We've got all of these different variants. The problem comes in this thing, the odds ratio. What is the chance of you being a migraineur if you have one of these SNPs or another one? And you can see that for all of the SNPs that we've found, the odds ratios are very small. They vary from 11% increase in being a migraineur to a 10% or 11% decrease in being a migraineur. And the way that the statistics work, we've found the big effect sizes. They're the easier ones to identify. So we'd need even more hundreds of thousands of people to identify many more. So in a way, I don't think from a kind of personalized medicine perspective, to know whether I'm 10% more or less likely of getting migraine is pretty useless information really, diagnostically. You might think, that's okay, we could add all of these up, and then we'd have a very precise level as to whether you're going to be a migraineur or not. No. If you read this paper carefully, what they say is the total variability in being a migraineur, only about 20% of it is explained by all of these SNPs. So at the moment, whilst we can get some clues, and those clues might be useful for biological purposes. So for instance, some of these SNPs, it was surprising that very few of them were SNPs in the genome that related or close to neuronal genes. Most of them were associated with nitric oxide signaling, and 30% of them were associated with vascular function. So there's been a big argument in the migraine field whether it's, it's a neurovascular problem, but whether it's a problem of predominantly neuro or predominantly vascular has been a series of hot debate. This may reignite that debate, because actually this data says that the SNPs that are associated with migraine, many of them are vascular. And so you could use that information to follow the biology. So it's good from a biological perspective, but it's pretty rubbish from explaining my or your risks. And as I say, I think it's not going to get better than this. This is the best data set we have. And I think for low back pain, given the heterogeneity of such conditions, I think it will be hopeless to try and expect this to perform better with other chronic pain cohorts. So, whilst I'm a bit negative about it, I would point out that it can reveal some interesting findings. This is actually not from a GWAS, this is from the candidate approach, and it's some data that's come recently from a colleague's lab, Dave Bennett's lab in Oxford. And so he's been interested in patients with diabetic neuropathy, and particularly the question, what makes about half of the patients with diabetic neuropathy have spontaneous pain and the other half don't? And the question is, well, can you identify who's who? And Dave's been looking for a long time for any marker that identifies who's a painful diabetic neuropathic patient, pretty much without any luck. But in this case, I think this must be telling you something. What he did was he took a couple of hundred of these patients, 111 of them had pain and 80 of them didn't, and then he looked for variants in one gene in NaV1.7. And what he found was that 11 of 111, which is a statistically much bigger number than 0 out of 79, this is a meaningful statistic, 11 of these people had mutations that affected the activation and inactivation characteristics of this NaV1.7 channel. So in a way, that's quite an intriguing finding. What it says is, if you're unlucky enough to get diabetes for whatever reason, maybe you need to have a series of unlucky factors to become a painful diabetic neuropathic patient. So maybe you need to have another trigger, which in this case, in a fraction of people, is an abnormal sodium channel. Now, the abnormal sodium channel is nothing like as penetrant as the cases I showed you earlier. So the implication is that outside of their diabetic skin, in their proximal body regions, these people have normal pain sensitivity. They still have an abnormal genome, their 1.7 is mutated everywhere, but in their short axons, they don't have sensory abnormalities. It's only when they have diabetes plus a funny NaV1.7 are they more likely to feel pain. So I do think this approach might be useful looking forward to help us unravel some of the characteristics that contribute to chronic pain states. So I don't think it's a worthless exercise. I just think the traditional GWASs may not be as informative as we first thought. Okay. I'd like to shift now to this second topic, which is a slightly different one, and it's more focused on animal models, and that is how changes in gene expression occurring as a result of some pathology, like arthritis or some neuropathic condition, how they may contribute to the pain that emerges, if indeed the animals do feel pain. So, let me start by a couple of bits of evidence that I think the place to look for pathology is in the peripheral nervous system. And you've seen this argument enunciated several times, but I'd like to make it very clearly, because I think it's an important argument for the pain field as a whole, whether you're a clinician or a preclinical scientist. So this is how important are abnormalities in the peripheral nervous system from a clinical perspective, and I mean use that term loosely. Clinical meaning the painful condition. And I would argue I would argue that There are pathologies in the pain signaling system throughout the neuroaxis in many conditions. And of course, in theory, you could treat pain by correcting those abnormalities anywhere from the periphery to the highest central levels. But I'd make the point that an attractive place to work would be in the periphery, because in many, not all, but in many chronic pain patients, peripheral nerve activity seems to maintain the abnormal sensibility that they have. So there's two bits of data on this slide. This one I think Tony alluded to. It's the outcomes of knee and hip replacement from people with chronic OA, and it's pretty similar for RA patients as well. So again, I'd point out that certainly if you come from a fairly impoverished part of the world like the UK now, you don't get a joint replacement for 20 years. So you are a chronic pain patient by the time you get this, and you've had plenty of opportunity for the pain to become centralized, burnt into your CNS. But actually what happens is, well, maybe we can look here, the large majority of these patients get an improvement, often a very good complete improvement in their pain on joint replacement. Some of them don't. There are 20% who seem to get no benefit, and we shouldn't forget those. I mean, they presumably don't have a pathology in their peripheral nervous system associated with a joint. So this is, again, evidence for a chronic drive. This is data that Per alluded to from a study published a couple of years ago with a small number of peripheral nerve injury or diabetic symmetrical polyneuropathy, distal polyneuropathy patients, in which all of their sensory abnormalities, that's the pain and the ongoing pain and provoked abnormalities, all resolved 100% transiently to local anesthetic nerve block. And for what it's worth, that's the data here. A much smaller effect was seen with intravenous block, and I think we can learn then that if you're looking to answer this question of peripheral versus central maintenance of pain, intravenous lidocaine is probably not a very good test. You probably can't give enough lidocaine without killing people to get the full efficacy, whereas the nerve block obviously can have a very, very high local concentration. So that is a small sample. I mean, that's one of the problems with it. But I think this is another very important paper that I alluded to, and it comes from Marshall DeVore. And he convinced some surgeon, I don't know, it is somewhere, to apply local anesthetic to the DRG intrathecally to the DRG and spinal cord in patients with phantom pain. And he had quite a lot of argument. So what he found was that all of the patients had a relief of their pain from central block, this spinal block, whereas very few of them had very good relief from trying to inject the stump itself. So his argument was there must be a peripheral generator in the DRG that is sustaining the pain. And it's true that he couldn't completely disambiguate DRG from spinal cord, but he had quite good evidence on doses and location that this was primarily a DRG effect. And so this will be another important piece of evidence that for these patients, a peripheral input is a key driver of their pain. So I would argue that if you're looking for mechanisms, if this is true, that these peripheral pathologies are driving a sustaining neuropathic pain in patients, well, let's find out what causes those in animals, and then we can ask whether that's true in patients. And that does seem to me a very rational way of doing sort of translation. If you have a clear idea about what you're looking for, you can study that legitimately in animals in the hope that it will translate. So one other point, again, I don't have to make this, Per's already made this, that there are many things that cause neuropathic pain, including traumatic nerve injury. I think it's clear that that is often painful. The problem is that it's often painful when you do it, but it's not a major cause of neuropathic pain. And yet, I would say 90% of what we know from animal studies has studied only this kind of nerve injury. And that's a bit of a failure on our part, because we do have models of all of these other things. In some cases, they're not very good models, but it would be worth us trying to, that is going on. We are trying to extend the range of models that are commonly studied now. I mean, a good one will be neuropathies associated with chemotherapeutic agents, because we know the time course, we know the doses, we know what happens when we give those agents to people, that we cause, iatrogenically we cause pain, and I'm sure we can model that very accurately in animals. But it is true, as Per pointed out, and I would reiterate, that the mechanisms I'm going to tell you about are particularly relevant to traumatic nerve injury, and we don't quite know whether they hold for these other conditions, which is a bit of a $64,000 question. Okay, we know what the sensory abnormalities are in these neuropathic conditions. They involve a lot of spontaneous pain, as well as abnormalities in many patients to applied stimuli, sort of mechanical stimuli or thermal stimuli. And we're quite smug. I mean, on the bright side, we are able to emulate some of those sensory disturbances in animal models. And the thing that we've done, we've been quite ingenious in damaging major peripheral nerves in an animal, usually the sciatic nerve, not exclusively, usually a rat or a mouse, not exclusively. And the trick has been to partially denervate a major target, such as the hind limb, by damaging some but not all of the fibers in a peripheral nerve in ingenious ways, like tying off different branches or self-strangulating the nerve with an inflammatory gut ligature so that some fibers undergo Wallerian degeneration. And the idea is at the site of these lesions, some nerve fibers, some axons are interrupted, and they will undergo Wallerian degeneration. And for what it's worth, those animals do show similar changes in they become hypersensitive to a whole number of applied stimuli in a way that is qualitatively not dissimilar to what can be seen in some patients. So they have the same kind of sensory abnormalities. The assumption is that they would have spontaneous pain. And if you notice the slide I showed you from Troll Jensen's nerve block, he did say that spontaneous pain and provoked pains all went down at the same rate. So, I mean, you could interpret that as saying that whatever the pathology is, it looks to be the same underlying pathology for spontaneous pain and provoked pain. If that was true, that would be good for the animal experiments. It would be one reason why, even though we're only able to measure provoked pain, we're still studying the same mechanism that's associated with ongoing pain. But I completely agree with Per that as a community, it is our job to try and improve on these outcome models of neuropathic, of pain that may relate to spontaneous neuropathic pain. And it's probably worth saying, I mean, it's not that people have just sat around doing nothing. A whole bunch of things have been explored often with limited success, but people have tried to look at catwalk, locomotor behavior, they've tried to look at facial expressions, some of these burrowing studies, that there are a number of things that have been explored, nothing yet is looking great. I said I'd tell you what I'm trying to do. I came across this company who, as far as I can see, most of the time, they work for Google or the Ministry of Defense on dodgy face recognition software from videos. So they're trying to spy on us all the time. But they've gone into business trying to spy on animals. So they've made, with our genomics unit in the UK, a system in which you have a home cage which has RF antennae in the floor of it. You implant a little RF chip, the kind of thing that supermarkets use to keep track of their tins of tomatoes. You input that into the animal, and the animal is then monitored by an infrared camera 24 hours a day. And what they can do when they have this system is, if I can... so I don't know if you can see that the computer is able to monitor each individual animal as it moves around the cage. It knows who's who because of the RF chip. The dodgy bit of it is that they're analysing from the video the behaviour of the animal, and they're able to calculate an increasing number of complex behaviours that the animals perform in their natural home environment without any intervention, no observers, no handling, no nothing. And so the hope is, I mean, no one's ever studied it in the pain context before, but I'm very hopeful that we might see some behavioural consequences.
Do you have a diagram of
You can mix them or you can do them as you like. So so far, all of the development has been done with a genomics unit who've been studying, they've been using it to screen genetic mutants. And so this one is a brilliant one. They've published a little bit of this. So with a single animal, this is the animal's behavior over 7 days. It's a single animal. You can see there's the light-dark cycle and the difference in activity. You can pick out lots of subtle things that I'd say you'd never pick up in the kind of testing that we normally do. So here, the lights go off, but before the lights go off, this animal knows that the light's going to go off and it starts increasing its behavior, which is documented here. And in fact, they noticed that with one transgenic strain where they had one knockout mouse with 2 normal mice, the phenotype of the mice were completely normal in every acute test that they had done, not pain, but other tests. But in this phase, this mouse was socially isolated from all the other mice. The other mice ignored it for some reason. So obviously it's jam tomorrow, but I'm hoping that some of these more complex measures might be useful for us to develop the kind of outcome measure that even Per would be happy with or would tolerate. Okay, so that's, I think there are some things going on. Back to mechanisms, because, as I said, I think the other bridge between the animal and the human work is to understand the mechanisms that are operating in the animal and then ask if they are the mechanisms that are driving neuropathic pain behavior in the people. So, I've told you that the interest is on the peripheral nervous system. So what are we doing to the peripheral nervous system that's causing the pain? Well, there's been quite a big argument. So what we've done is, if you imagine this is an illustration of all of the kind of lesions, we have the sciatic nerve projecting through a couple of posterior roots, and we've damaged some of the nerve fibers in that circuit, either in one root or in a variety of ways. The question is it the damaged nerve fibers that are causing the abnormal behavior that we can measure, the stimulus-provoked behavior, or is it the spared nerve fibers that are still intact but have changed their properties so that they're driving the behavior? And rather surprisingly, we don't have a clear answer to that question. We have data both ways suggesting that it's the damaged nerve fibers, and we also have some data suggesting that it may be the spared nerve fibers, and the interpretation is difficult. I'm just saying that we're not 100% clear. So mechanistically, what we're doing is we've got some major piece of tissue supplied by several nerve roots.
Can I just add one thing,
Yep.
Stephen? From the clinical perspective, again, it's not the spared nerves that hurt, because the pain is projected into the innervation territory of the injured nerve, not the non-injured nerve.
But these are, a single nerve has some spared fibers and intact fibers within it. So both the damaged nerve fibers and the ones that are left have the same innervation territory, the erstwhile innervation territory, yeah. So we're making some lesions somewhere in this trunk, and the idea is that the lesioned axons degenerate. We have a reduced innervation, so there's actually, it's quite easy to understand why you might have a loss of function in these traumatic nerve injury cases. You've lost most of your sensory nerve fibers, but actually you still have some gain of function. That's the pain and the hypersensitivity. And so the issue is, what is happening that's causing this pain and hypersensitivity? And I would say at the moment we have sort of some understanding, which I will try and elaborate on. One is that, the damaged nerve undergoing Wallerian degeneration, that is a war zone. At least for several weeks or months, there are the invasion of hundreds of macrophages which are eating up the damaged nerve fibers and releasing God knows what. There is, secondly, a loss of the trophic influence coming from the targets. We know from a lot of neurobiology that sensory nerve fibers interact with their target fields. They get trophic factors from their target fields. Obviously, if you're disconnected from your target field, you lose those trophic factors. So we have two major causes. We lose retrograde trophic factors, and we have the presence of a whole number of injury fibers. And particularly, if you're a spared nerve fiber, you're running through a degenerating nerve, which is making bucket loads of chemokines and cytokines. Anyway, something is arising out here, and I'll tell you maybe what it is. And the secondary consequence is that that changes gene expression in the DRG, and as I'll show you, a critical event in animals, and I've already told you from humans, with some kinds of injury, a critical event is the emergence of ongoing spontaneous activity, which is underpinned, we believe, by changes in gene expression. And it's this event which is driving all of the central pathology. There is a lot of central pathology, but it's being sustained by this abnormal peripheral pathology. So this gives me an excuse to try and tell you a little bit about this, but mainly, what are these genes that are altered when nerves are damaged, and how do they contribute to the ectopic firing that emerges? So, first of all, to say just what happens in the peripheral nerve. Here's some nice data from Beth Morinson at Hopkins. She did a very simple experiment. She made one of these kind of rat models where we partially injure the sciatic nerve, and she counted the size of Remac bundles distal to the injury. So the Remac bundles are the non-myelinated Schwann cells and all of the C fiber axons that they contain. And what she found was that the size of the Remac bundle, as you can imagine, went down from an average of about 4 to an average of 1.5. So that means that there are individual C fiber axons that are projecting through Schwann cells that are reacting to the that they are responding to the axonal degeneration that's going on in some of these neighbors. Individual Remac bundles will have intact and damaged fibers. And is that relevant? It's relevant because the Schwann cells that are subject to this injury start making lots and lots and lots of things like NGF and many other things. And that may have an impact. That becomes a sensory message on the intact fibers who are exposed to a lot of these signals. Does that matter? It matters because the DRG responds to these positive or negative signals, loss of trophic factors or the addition of injury factors, by quite a large shift in gene expression. And this experiment has been done a lot in the last 15 years. It's actually a little bit confounded, but I like this early version of the experiment that looked at gene expression changes on a fairly small, custom-made sort of AFI chip for just a few thousand genes. And obviously you won't be able to read the details here, but what they're showing are individual genes that are dysregulated, either upregulated or downregulated at different times from, I think, 1 day to 4 weeks after nerve injury. And the important point about it is that many of the genes that control the excitability of nociceptors are dysregulated. So some of the genes that encode for transmitters that are released from nociceptors with activity are changed. So some neurotransmitter genes are downregulated, some of the peptides like substance P. In other cases, we get a de novo expression, a new expression of factors that act as neurotransmitters like BDNF. Some of the receptors that are expressed on primary sensory neurons, both in their peripheral terminals, which have been damaged, and central terminals, which are important for regulation of sensory inflow, are also dysregulated. And that includes TRPV1, used to be called VR1, P2X3, the mu-opiate receptor goes down in primary sensory neurons after nerve injury. That may account for some of the loss of sensitivity in these models to opiates. And then I think from the context of what I'm going to tell you about activity, ectopic activity, clearly a change in ion channel expression is likely to be crucial for changing this sensitivity, the excitability of these nerve fibers in a damaged nerve. And actually you can see in this block, they only have, Alain told you there's a lot of ion channels, only a few are on this chip. This blue blob here is the alpha-2 delta-1 subunit of a calcium channel subunit, which is known to be the binding site of gabapentin. So it's interesting that that is dramatically upregulated, as Tony mentioned, in neuropathic pain models, and may account for some of the actions of gabapentin in these models. Nonetheless, this, it's easy to see how these hundreds and hundreds of gene changes, changes in gene expression could contribute to the abnormal function of sensory neurons. I'm going to finish in a couple of minutes. One thing to bear in mind that has happened recently is that we think of the DRG as an homogeneous structure. This is it, it's a DRG from an animal stained with some green neurofilament stain, or beta tubulin. But actually, and most of the data we have on gene expression change comes from just taking out ganglia and analyzing gene expression in the whole ganglia. But we know that the ganglia is quite heterogeneous. It's made up of different types of neurons and lots of non-neuronal cells. And actually, one important point that until recently has been overlooked is that when you damage the nerve in any way, you get a huge infiltration of immune cells into the DRG. So if you simply take out the DRG and measure some gene expression, of course it changes. It changes because neuronal genes are going to be diluted by RNA coming from non-neuronal cells, and the expression of factors that we are interested in may go up because it doesn't go up in neurons, it goes up in these non-neuronal cells. So I think for what it's worth, you have to be very careful, and one of the points I wanted to make is that we are entering a new era now where people are no longer doing expression studies on whole tissues. They are trying to isolate the tissues that they're interested in. And so it's quite easy by a variety of sorting techniques. If you take a whole DRG and dissociate it, you find out that only 20% of the cells are neuronal. The other 80% are non-neuronal. It's quite easy to purify these cells and then either do sequencing on pure neurons or on individual neurons. And in fact, I was going to show you some examples. If anyone's interested, I would encourage you, I can give you some literature. It's quite easy to look up gene expression in your favorite cell type for any gene. And you ought to do it because if you make claims, if you plan experiments that depend critically on the expression of a particular gene in a particular cell, it's pretty easy to find out whether it's there or not now. There's a huge bank, so there's an effort called Blueprint, which is looking at every kind of immune cell in blood, doing genome-wide profiling under a number of conditions. There's lots of neuronal data. There is data for astrocytes. There's data for microglia, which I'll talk about tomorrow. What is being expressed, at least under normal conditions and some pathological conditions, is actually very accurately described now. And if you ignore that data, I mean, one, you're asking for a hard time from reviewers. I mean, I often review papers that tell me the secret of neuropathic pain is something that is not actually expressed in neurons. So I think it's well worth looking at. Anyway, let me quickly finish. Why are these gene expression changes important? They're important because they lead to this abnormal ectopic activity in many sensory nerve fibers. Yes, lots of myelinated fibers, and with a slightly still poorly described time course in unmyelinated fibers. So many fibers fire either tonically or in little bursts, zip, zip, zip. And this ectopic activity, we know behaviorally, is correlated with the emergence of these abnormal sensory responses in animals. We know that they occur in both the damaged and the intact afferents, and we know that they're essential for the behavior of the animals. If you give local anesthetic, if you repeat the experiment I showed you from Marshall DeVore in a rodent, you block the abnormal activity. So a critical question for us, and I think for the clinicians, will be, can we identify what is the molecular mechanism, the mechanism of abnormal gene expression that is driving this ectopic activity? It's causal, so it's not been done very much, but there are some very nice experiments done by Pat Wall, actually, 20 years ago, putting TTX on a single nerve through a micropipette to block nerve activity, and then looking at gene expression changes, and you don't get spontaneous activity, and you don't get, they didn't measure many things, but you don't get these changes. So these are changes that are caused by a chemical stimulus. We don't know what that is. I've got quite a bit of evidence now. Where's Michaela? That might be IL-6, seems to be a good candidate, but that is triggering these changes in gene expression. So what is the key gene or genes that are causing ectopic activity? Well, if you read the literature, there's quite a lot of choice, but I would say we still don't know is the short answer. There's some studies out there saying that it's this sodium channel, that it's a strange case. This sodium channel goes down with nerve injury, yet nonetheless antisense against the sodium channel was claimed to reverse neuropathic pain. Don't believe that. And if you look at a NAV 1.8 knockout animal, it has completely normal pain-like behavior, if that's what it is. So whatever's causing our behavior, I don't think it can be that. We did some experiments a few years ago with John Wood, in which we looked at different sodium channel subunits. The only one that goes up after injury is this one, NAV 1.3, and we thought that's it. NAV 1.3 is the secret of neuropathic pain, until we made a knockout that had exactly the same sensory abnormalities, so I don't think it's NAV 1.3. You heard from Alain that there's many potassium channels that go down with injury. They're all great candidates. We published a paper a couple of years ago saying that this one might have a role. It's one that from one of these association studies in humans was suggested to be associated with a kind of low back pain phenotype in patients. And we provided some evidence in DRGs that it was present, it went away with nerve injury, and it seemed to underpin some of the ectopic firing, at least the firing that occurs in large fibers. And so we thought that was the problem. But actually, we don't really know what causes this ectopic activity, and so I say that was the $64,000 question. And I'm going to finish there. I'm going to talk about sex differences tomorrow and some of the neuroimmune interactions that arise centrally as a consequence. But okay, sorry about the time. Okay, thank you.